I think we're going to just wait for a few more minutes. I think people are still coming up to maybe a few more minutes, like 2-3 minutes, because people are still almost finishing up their lunch and heading towards here. So after a few minutes, we will start soon. Okay. All right. So alaikum and welcome again. So while we're waiting for people to join us, any question from last two sessions? So I got pretty good idea about like general genes, oncogenes, and tumor suppressor genes, and what are they, and how they play a role in signal transduction, right? Because they're all the components of signal transduction, and that's how they modulate the activities of cell, basically cell cycle. Okay, so we're going to continue on the same theme, the cancer genetics. So we talk about the cancer genetics, okay? And last time we talked about genes, right? Oncogenes and tumor suppressor gene. So they play the, of course, they play the very important, essential role. But there is a thing called epigenetics. Okay, have you heard this term before? Epigenetics. Do you know what it means? Okay, you know what it means? Okay. Well, epi means above, right? And genetics is genetics, right? So anything, this is like above genetics. Okay. So to give you an idea, you may have seen that this is actually babies born by a mother, right? So a mouse has usually mouse has like litter. We call it litter, and they have like 6 to 7 babies, right? So, and they are all born almost like same time. But if we can see that they are born, but they have different color codes, right? Like brown, light brown to almost like darkish or blackish brown, right? So they vary in color. Although they are like color codes of genetically identical. Genetically they are identical. They're their gene whatever they're expressing is identical, but they have different color codes, right? Similarly, you may have seen, people who are twin, right? They're pretty much identical in their looks, right? But something is different. Like, she's has red hair. She's a red hairs, and she's a blonde, like a golden hair, right? So their hairs are different. So I did identical twin with different hair colors. Okay. And sometimes what happens? So these are two different individuals. In this case, this dog, it has two different colors of eyes, right? One is kind of blue, and other one is like kind of greenish, brownish, greenish, right? So within one animal, there are two different colors, right? So all these, and then not only that, within one eye, they are like different colors. Like outside it's like greenish and then brownish, right? The spots. So within an eye, there are different colors are expressed, right? So all this is because of epigenetics. Okay, so what happens that we know that we all are derived from one single cell, right? What happens that when sperm and egg they fertilize, they make a zygote, first cell, right? Okay, so this how our life begins. After that, these cells divide and divide and divide and divide and differentiate, and then we become like this, right? Our individual. So when we have one cell, so this is like it expressed all the genes which are required. Most of the genes, all the genes which are required there, they are get expressed. Okay, so this is kind of, we call it totipotent. So this is like a total expression. So this cells has the ability to self-divide, express all the genes, and then as the cells grow, after certain time, then you probably know that the cells that it differentiate into layers, right? Ectoderm, mesoderm, and endoderms, right? So those three jumps layer, right? And then from those jumps layer, then it further divides into different organs. Like some organs they are derived from mesoderm, some are derived from ectoderm, some are derived from mesoderm, right? So as the cells are dividing and differentiating, so some genes which are not required, they get silenced. Okay, the only genes which are required, then they get expressed. So then we get totipotent, then we have like pluripotent, whereas there's still a lot of genes are expressed, then multipotent, which like a cell which drive different kind of cells, and then unipotent. Unipotent when these cells finally kind of determine its fate, that this is what is going to be like a muscle cell is going to be a muscle cell, so it's going to just express only the genes which are required for the activity and function of muscle, right? Always derived from the go back, they are derived from the same cell, right? Same as our blood cells. If you go back, they derive from the same cells, but then because of the differential gene expression, they become blood cells, right? So that's how the what happens, right? So epigenetics, as I said, mean above or in addition to genetics. Okay, so we know that how do different adult stem cells. You heard this term before, like stem cells, right? So these are the cells which can be differentiated into different cell lineage. Okay, and then we know that myoblast. So myoblast is kind of stem cells. They can only, but they are like already. So they are here. Somewhere here, like unipotent, right? Because they are going to be muscle cells, right? So somehow they are they are derived from some cell, and they kind of differentiate into myoblast, and they're going to make uh, muscle cell, right? Same as keratinocytes, they make skin cells. Hematopoietic cells, right? As we know that we have hematopoietic stem cells, right? So the pluripotent cell diverge into hematopoietic lineages, and then the hematopoietic cell make all kind of blood cells, right? So but all have identical DNA sequence. So all cells, they still have the same gene is not changed. Everything, muscle cell has the same gene exactly what our eye cells has, or heart is muscle anyway, so adipose cell has, right? Or nerve cell has. Okay, but nerve cell is nerve cell because the gene which are expressed in nerve cell, they are not expressed in, not in, there will be some gene which has similar function, will be similar, but then there will be genes which require for the function of muscle, they'll be expressed in muscle, And the genes which are required for the function of nerve cell, they will be expressing nerve cell, right? So that's how they get differentiated, right? So by definition, an epigenetic trait is a stably inherited phenotype resulting from change in chromosome without alteration in the DNA sequence. So there are multiple ways, there's a definition of epigenetics. So what it means, there are stable inherited phenotypes. So these are the phenotypes that can be inherited, right? Once the change is made, so there is no change in DNA sequence, there is no change in gene, because how the gene is regulated, then that change and we will go through what are those changes that can be transferred from one generation to other generation, right? Okay, now the US National Institute of Health, NIH, stated that epigenetics refers to both heritable changes in gene activity and expression, which is like in the progeny of cells or individual, and also stable long-term alteration in the transcriptional potential of a cell that is not necessarily heritable, right? So what it means is that they are epigenetics involve both, right? Some genes, some mechanism or some epigenetic processes can be inherited to certain generation, but they can be reversed. Okay, so I mean, for example, common example, you see that sometimes you say that a father has some people have they are left-handed, right? And then their kids also, all of kids are right-handed, but one kid, it's like left-handed, right? So, and genes are still the same. They all came from the same parent, right? They still have the same gene, but because they're what we call epigenetic changes. So that lead to certain behavior, like you do so, or you egg something, or you just gesture, like making the hands like this, like you make your father is making. So all these things somehow get imprinted in the gene, right? So it's not is there's no gene for like different behavior, but it's the epigenetic changes which actually determine different kind of different behaviors which tasks can be inherited, right? And after a generation, they can be disappeared because those genes are those changes in DNA structures are changed, then they will be disappear, right? Or the epigenetic modification are reversible, right, and can be removed from genes and chromosome without leaving behind any permanent changes to the genetic material, right? So these are various definitions. So in short, epigenetic changes are the changes on genetic on chromosome, basically, without altering the gene structure. So it's not going to be any mutation or any deletion of any gene or activation of gene or hyperactivation of gene, right? It's just that there are mechanism which changes the structure of chromosome, right? And these can be inherited or they can be non-inherited. Okay. So there's one famous example here that is Hunger Winter in 1944. So this is a place in Holland, right? So remember, like in the Second World War, what German did, they blocked the food supply to Dutch people, right? As a result, the people were on ration, right? So they were taking less calories, right? A normal person usually take about like a 2,000 calories. So their calories was reduced to one fourth, like 500 calories per day for about 4.5 million people, right? And then what happens that if you have less intake of food, obviously it's going to affect your activity, your growth, right? So children born and raised in that time were small. They have like a short height, and they were susceptible to many diseases like edema, anemia, diabetes, and depression because of the caloric restriction. But what happens that the Dutch famine birth cohort study showed that the women living during this time, right? So the babies who was born at that time, their children 20 or 30 years later, right? And those children still like have the same features what their parents had 20 years ago, though they were living in a normal dietary status. So what happened that because diet play, and we'll show some example, diet also play a very important role in for epigenetics. So you probably heard the term, you are what you eat, whether you believe it or not, but this is very true because if you eat things which are junk, that might affect some gene activity, and that can lead to diseases and things, not only to you, but it can pass on to next generation too. So some people on a diet like this which induces diabetes or obesity. Okay, but if it is kind of because of that dietary condition, if it is get imprinted in the gene, then it means they're making their next generation susceptible too. So it can be inherited, but it's not permanent. It can be reversed. Okay. So in order to understand the epigenetics, we have to understand the structure of chromosome, right? And I'm sure all of you have probably seen that how the chromosome is organized, right? So the basic structure, double-stranded DNA, right? Everybody knows that we get, DNA is double helix structure, A pairs with T, G pairs with 3, right? And they are complementary bases to each other. And this DNA structure, actually, it can wind it on histones, right? So like it become like smaller. So all this like is like a thread. You put a thread on a ball, so you can wrap so many layers of the thread on a ball, right? So DNA become condensed, and all these they become like further condensed. So these like we have a histone where DNA is wrapped on, and these are then become like very condensed. They make chromatin fiber, and this is become more condensed, right? And it's condensed, further condensed, and they make a chromosome. So these are like tightly, very tightly packed DNA. Chromosome is nothing but very tightly packed DNA, right? So this chromatin, what we say is basically is a term used to describe DNA complex with proteins, mainly histone, which are found in eukaryotic chromosomes, right? And chromatin can be like divided into, actually they are divided into two, like euchromatin and heterochromatin, right? Now the difference is that euchromatin is less condensed. Okay, it's less condensed, and because of this, they are transcriptionally active DNA. They are rich in genes. So all the genes which are transcribed, they are usually localized in the euchromatin. Okay, and they are less condensed because they are weakly associated with histone. Okay. They undergo replication during the early S phase. We know that cell divide, and before that it prepares it replicate DNA. Okay, genes may or may not be expressed depending on the cell type and functional needs of the cell. And it's controlled by reversible covalent modification of DNA, right? Okay, so this uh, uh, chromatin condensation, which is controlled by covalent modification, and these are what? DNA methylation and histones, modification of DNA and histone. In case of DNA, we talk about this, DNA get methylated. In histone, there are other covalent modifications like acetylation, methylation, phosphorylation, and ubiquitination. So then histone can be modified in a lot of different ways, right? And then these regions also code for non-coding RNA, right? So these are the RNA. We know that RNA make use for these are messenger RNA, which we have different kind of RNAs like ribosomal RNA involved in protein synthesis. We have tRNA, which transfer the protein, amino acid on the ribosome. We have ribosomal RNA, protein machinery, and then we have mRNA, which carry the message for the synthesis of protein. In addition to those RNAs, we have other RNAs. And some of these RNAs, they also come from, they are non-coding, so they're not coding anything. They are involved in RNA-associated silencing. So what they do, because they have they are complementary to certain genes. So these RNA bind to the mRNA and degrade the mRNA. So the message is lost, right? But sometimes they also activate, and depends, sometime, and we'll go if we get time, we will go touch on different RNAs. Okay, so on the other hand, the heterochromatin, they're tightly condensed. You see how tightly they are. So when they are tightly condensed, and they are tightly condensed because they are condensed with the histone protein, and because they are tightly condensed, they are transcriptionally inactive, right? They are mostly located in the regions of genome that undergo replication during the late 8 phase of the cell cycle. Right? Contain relatively few genes, which rarely undergo expression because they're so tightly condensed. They're still people don't know why this DNA is there, which is not going to transcribe, but it's still maybe they're to support the structure of the chromosome, right? It is characterized by low acetylation, right? And low acetylation level of histone tail N in a high DNA methylation. When we talk about, because DNA methylation contribute in condensation. Okay, it's recognized as a structure that protects the ends of chromosome. So it's and it determines the separation of chromosome to daughter cell during the mitosis, centromere. So this is her example. So this is like a euchromatin, and euchromatin sometimes is when it is not expressed is converted into heterochromatin and then is silenced. When it need to be transcribed, then things happens on the histones like acetylation. When histone acetylated is acetyl group is negatively charged, so they get heavily acetylated. So negative charge repel each other, so DNA get extended, right? So the heterochromatin become like euchromatin, so it's open now. So genes are open and then it's transcribed. So acetylation is a process where acetylation of histones which involve in gene transcription, right? And then you probably may not be able to read or see this thing. It's just the same thing that we have DNA and all that. So it just shows that there are a lot of modifications happens, and there's like listed here is like I mean I can read. It's like there. Okay, my class is not really strong. Okay, so there are like all these processes. There's like acetylation, phosphorylation, that happens along the chromosome. Okay. Now, epigenome. So, like we know, genome is a total gene study of total genes. So, same as sum of all the natural covalent modification of a chromatin is called epigenome, right? And when we cause chromatin, and again, as I said, chromatin means DNA and histone together, right? Okay. Epigenetics, chemical alteration of chromatin that influences gene expression. So again, the epigenetics is the alteration of chromosome or chromatin, and we know that so it's involved both DNA and histone. Okay, without changing the sequence of DNA nucleotide. Okay, so there are like there are three different classes of signals operating in a concerted manner, create heritable epigenetic state. Like so, we have like epigenetor things we stars a trigger. Again, and then we have a epigenetic initiator, so which determine the precise location where this epigenetic, or chromatin is going to be modulated, right? And then we have epigenetic maintainer, which kind of keep as an internal signal, and then once the epigenetor change, it could be an environmental thing, it can be food we eating, it can be like so. These are like epigenetor. Then because of that, it find a place where it's going to modify the chromatin, and then once it's modified, then we have a certain enzyme which can remember that place. So when you need DNA duplicate, it goes again and then modify that part of the chromatin. So they are called epigenetic maintainers. Okay. Now, type of epigenetic changes, as I said, it's involved both chromatin, DNA, and histone. So it's mostly is DNA methylation and histone modification. And histone modification, I said there are a number of things happens to histone: acetylation, ubiquitation, phosphorylation, a lot of things. So these are the those processes they are involved in epigenetic changes. Okay, so basically, what we say that we say epigenetic change, right? So epigenetic code. So epigenetic code is the DNA methylation, is a sum of DNA methylation and histone modifications. Okay, so again, I've shown you this example in a different format that we have our chromosome here, which undergo acetylation. Okay, so acetylation actually opens the chromatin chain, and it also goes like in methylation process of deacetylation, and then so it's like open for transcription, or when it gets methylated, then it gets condensed or deacetylated, right? So methylation and deacetylation cause condensation of chromatin, whereas acetylation causes or demethylation causes opening of the chromatin, right? Now, we're going to talk about first the methylation process, right? And then we will talk about the histone later on. So DNA methylation, what happens that the residue which get methylated is cytosine. Okay, so cytosine is involved in methylation. So, and this we know that cytosine bind to guanine, right? Okay, complementary. So they get methylated across, on the other, both strands get methylated, right? Um, so DNA methylation and histone modification help to compartmentalize the genome into domain of different transcriptional potential, right? What it means that different chromosome or different chromatin have different transcriptional potential, right? One which is open has a potential to transcribe. One which is highly condensed has a potential to not transcribe. So euchromatin is high in histone acetylation, right? Because acetylation is going to open. It's low in DNA methylation, right? And mostly the histone H3 and K4 methylation is involved. When heterochromatin is low in histone acetylation, is dense DNA methylation is all methylated everywhere, right? And histone H3 and K9 they are involved in the methylation process. Okay. Now, so this is, you probably have seen the structure of cytosine before, right? So it's a nitrogen base, and so it's get methylated. So S-adenosylmethionine, you know that methionine is an amino acid, and S-adenosylmethionine is the methyl donor. So it donates the methyl group, right? And itself is converted into S-adenosyl homocysteine, and this is DNA methyl transferase, which transfer the methyl group from S-adenosyl methionine and you get methylated cytosine, right? So, in addition to having genetic causes, cancer can also be considered as an epigenetic disease, right? So, regulation by genetics involve a change in DNA sequence, right? Where epigenetic regulation involves alteration in chromatin structure. Okay, so what would be the case? How epigenetics can be regulating the cancer. We know that genes are, for example, oncogenes need to be expressed, right? And to suppress a gene, oncogenes, in order for cancer, those oncogenes express, right? Or somehow the tumor suppressor gene gets suppressed, right? So the thing is that if the genes which are coding oncogenes, if somehow if they get hyperacetylated, right? So what happens? So they will be easily transcribed, right? Okay, and the thing is that the genes which are expressing tumor suppressor genes, right? If they get methylated, then what will happen? They will silence, right? So that's what happens. That some of the processes, these processes, so it's like DNA structure is not changing. So we know that cancer caused by sometimes oncogenes get activated by mutation. We change, like last time we had the examples, like we change a base and RAS protein, a normal RAS become like oncogenic RAS. Same as there's a frame shift, right? All the sequences change. Gene is overexpressing multiple copies. They're making too many proteins, or they're coding a protein which has higher activity, right? Or they're making too much protein, higher activity, or then the genes translocation. They're making a protein which is a new function, right? So all these are basically causing cancer, right? So there's involving change in DNA, right? Somehow the sequence is changing, right? But in this case, there's no changes in DNA structure itself. It's above the DNA structure, right? Epigenetics. So it's the methylation or acetylation. Okay, that would may result so in acetylation, transcription or silencing, right? Okay, so what happens that in DNA methylation, and I just there's a lot of mechanism, but if you understand the concept, then we're going to skip some slides so because I can still cover some of the topics which may be interest to you. So DNA is methylated, right? And as is divide, what happens that this is change is methylated, but this is not, right? So but there's like a DNA methylases, and there was an initiator which does the which involve the initiation when there's a new compound, new environmental factor or something, so they put down a new methylation, but methylation is already there, so there are methyl transferases, which remember where to methylate the DNA chain. So they go, and then once it's replication is complete, they go and then methylate again, right? So the two chains are separated, the complete chains are complementary chains are formed, and these then again methylated. Okay, so there's a lot of description, and I will give you the copies of these, after the lecture. Maybe, they will send you tomorrow or day after tomorrow. It's just a description that, in a normal cell, that 5-methyl cytosine is constitute about only 0.7% of all nucleotides. So in DNA, and affects 70 to 80% of CpG dinucleotide in the genome, right? In fibroblast, 98% of all methylation occurs CP dimers, right? So most of the time is like, when C comes with G. So these are what we call the C phosphate and guanine, right? So when they are two together, these are most of the sites when where the methylation occurs, right? Cytosine methylation is also observed outside CpG sequences, like in CPA or CPI, but they are not there. That or they happen rarely. Like, however, non-CpG methylation occurs relatively rarely in different differentiated cells. It does happen in embryonic cell. Embryonic and induced stem cells, as much as 5-methyl cytosines was found next to other nucleotide bases like adenine and in particular. So the cytosine in embryonic in stem cells, they may be present like in a CPA or CPT, but in the differentiated cells, it mostly happens in CPG, right? And this CPG, what we call is like we call CPG islands, because they are not only one CPG, but there are multiple CPG present together, right? So it gets hypermethylated, and there's called condensation of chromosome. So, and more recently, one is like 5-hydroxymethyl cytosine is also they also found, instead of only methyl cytosine, 5-hydroxymethyl cytosine. So previously they thought that this is just the oxidation of methyl group into hydroxylation because of the oxidative damage, but it turned out that there's some mechanism that 5-hydroxymethyl methyl cytosine turn out to be relatively abundant in some embryonic but not other heterochromatin nuclei of mammalian brain cells, about 0.2 to 0.6% of total number of nucleotides, respectively, right? So then I think it's become like, first they thought it's maybe the oxidative product, but now they think there's a real occurrence, that there's a mechanism by which cytosine can be changed into 5-hydroxymethyl cytosine. And in mammalian enzyme, they identified like this enzyme TET1 protein, which is a 2-oxoglutarate and iron-dependent oxygenase, which does the oxygenation. So to make the story short, that mostly 5-methyl cytosine, right? But 5-hydroxymethyl cytosine is also present, but very rarely. If it's present mostly in embryonic cells or stem cells, right? Now, gene silencing, right? So DNA methylation is a powerful mechanism for the suppression of gene. As I said, in a DNA methylated, it condenses, right? So there's a reciprocal relationship. What it means, DNA is more methylated, then transcriptional activity would be less. Okay, so there's like, if there's less methylated, mean transcriptional activity will be more. Okay, the methyl group, they do not affect any base pairing, but can influence other things. Because we know that in transcription, what happens, the DNA, when transcription occurs, you need a transcriptional factor. Transcription factor recognizes the binding element, right? Remember, there's a binding element, transcription factor come, the promoters and all that. You probably heard from microbiology 101 or genetics 101. So when the DNA is methylated there, so it actually blocks the place where these can this protein can bind to DNA, right? So that's how they disrupt the transcription. So the strong effect of 5-methyl cytosine in mammalian promoter region suggests that DNA methylation inhibits transcription by interfering the transcriptional initiation, right? There are different kind of DNA methyltransferases, right? It's like DNA methyl transferase 1, 2, 3A, and 3B. DNA methyl transferase 1 their role is in the to maintain the original pattern of DNA methylation in hemi-methylated DNA. Hemi-methylated DNA means like when the DNA is methylated on both chain, when it replicates, two chains are separated, and then we get a new chain, complementary chain is built. So in this case, one chain is methylated, the other chain is not methylated, right? So this DNA methylase transferase 1, its job is to go and methylate the other newly formed chain. So it's going to recognize the hemi-methylated, and they are all complementary, so it's going to go to other chain and then methylate it, right? Okay, so here is this unmethylated DNA. This is fully methylated. So DNA methyl transferase. So what happens that let's try to so then the function of DN methylase 3A and 3B is for de novo. So de novo is like that when there's an initiator, there's a signal, then this enzyme methylate DNA, whereas DNA methylase transferase 1 is for the maintenance. Right? Function of DNA methyl transferase 2 is not clear. This enzyme is characterized by a weak in vitro DNA methylase transferase activity. Actually, it's catalyzed like tRNA methylation, right? As I just mentioned, the DNA methyl transferase 3 and 3B, they are responsible for de novo DNA methylation and therefore for altering methylation pattern of genome. So these are, the DNA methyl transferase is there. So whatever in our genome is methylated, when we get, our reproductive cells are produced, like sperm and our eggs are produced. So they kind of maintain that. So after egg fertilization, they remember those enzymes. Remember, we need to be, methylate DNA need to be methylated. Okay, but these enzymes, these are the one which are de novo. So any change, what happening to us, right? They will initiate that methylation, and then methyl transferases 1 and 2 will remember that. Sorry, number 1 will remember when the DNA get replicated, right? I don't want to go much in detail. It's just that establish and maintenance of DNA. So this is the both chains are methylated on replication. They are hemi-methylated. The other chain is not methylated. So it goes under further replication. So it's all this. We have a DNA which is kind of de novo methyl transferases methylated, and then maintenance will just maintain it, right? Okay. Now, in addition to DNA methylation, what we just described, methylation of 5-methyl cytosine, there are other kind of methylation, right? For example, what happens, the term DNA methylation is sometimes used to describe the covalent modification of nucleic acid induced by certain alkylating agents, such as, in diet, there's like dietary N-nitrosamine, which react with nucleobase to form methyl derivatives of O6-methylguanine. And this what happens that this is like a non-enzymatic reaction. So this compound, which is nitrosamine, usually produce when if your meat, if it's burnt, and if you eat that meat, so it is highly carcinogenic. So eating a burnt meat is has a lot of this N-nitrosamine. But what happens most of the time, it's going to modify the guanine, and then it's going to do the DNA damage. But we have a good repair system. So most of the time that happens and DNA get repaired, right? It's only that if there's something wrong with your DNA repair enzymes, if somebody has some kind of mutation in those enzymes, if those enzymes have a problem, that's what happens, that they are very susceptible for all kind of their DNA is susceptible for all kind of chemicals, because their repair mechanism is not that efficient, right? Okay. Effect of methylation on gene activity. So what happens, and I want to just maybe describe like this. What is it? I heard something. Oh, somebody outside. Okay, sorry. Okay, so as I explained you, like we have this promoter. Okay, so if promoter is methylated, So what happens when transcription factor is going to come here? There is no methylation, right? Transcription factor comes, finds place, sit there, and start transcribing. Okay. Now this site, which is a promoter site, is methylated. So transcription factor it comes is not going to find its place. So what is gene is silenced? It's not going to bind to where it's supposed to bind, right? So there's one mechanism. Other mechanism that we have various methyl binding proteins, methyl cytosine binding proteins. So it's binding, and these proteins they actually come and put a cap. Okay, so it's the same thing that your transcription factor comes, it's not going to find the place and is not going to start transcription. Third mechanism is that this methyl binding protein, methyl cytosine binding protein, they attract other protein, okay, and make a protein complex. Okay, now the protein is usually attract is also a histone deacetylase. Okay, and you know what histone deacetylase does? It's remove the acetyl group. So when you remove the acetyl group, DNA is going to condense, right? So that's what happens. This methylation is blocking the transcription, and when it's recruiting the histone deacetylase, then is DNA is chromatin is also going to condense. So these are multiple mechanisms through which methylation of DNA contribute in silencing the gene. Okay. DNA methylation in cancer-related disease. So methylation pattern reading. So what happens that the promoter usually these get hypermethylated for genes against background global hypomethylation. Okay, so it's interesting that if you see in cancer patient when they see the whole when they look at the whole genome is actually hypomethylated, but then there are regions which are very methylated, right? So the thing is what happens in carcinogenesis is that the regions which supposed to be methylated and non-methylated, so they are easy to break down. So that's also contributing to carcinogenesis because DNA is break down, joined with other, the genes can move around, and then the genes which are hypermethylated, they are silenced. So it's like a multiple mechanism that the methylation play a role. So there's a characteristic of carcinogenesis is like overall it is hypermethylated, but they are local, there are specific regions which are hypermethylated. Okay, so the cancer cell genome is characterized by a decreased level of cytosine methylation relative to normal cell. Okay, so as I said, lower level of DNA methylation in this region increases genomic instability, thereby promoting the development of cancer. Proper hypomethylation is associated by region-specific DNA hypermethylation, particularly typical of the initiation stage, but also influence further stage of tumor development. So, okay. So in human prostate cancer, like cell lines like PC3, I'm sure some of you are probably using some of these cells like GO145, LN, CAP, all these has an increased expression of DNA methyltransferase 1 gene. Okay, in comparison to benign prostate cancer. So that which kind of indicate that this methylation is probably involved in converting into a benign tumor into a malignant tumor. Right. Similarly, the genes like CDKN2A, which is a gene for cyclin-dependent kinase. Remember we talk about cyclin-dependent kinase is very important for cell cycle progression. And then VHL, I don't remember what the gene encodes, but BRCA1 gene we talk about, right? So these genes, what happens? Their promoters get methylated, right? And these genes are inactive. Okay, and then again, these are the some of the examples. There's some of the genes silenced as a result of DNA methylation. So these are the genes, right? There's their function and there's their tumor type. So it's just example, just to show that it's not only one or two genes, but there are a number of genes that undergo into this process, right? Yes. Yes. There are people working on it to inhibit the methyl transferases, and there are all kinds of attempts. And we'll talk about a little bit more, down the lecture, right? Oh, here it is, anti-cancer drug targeting DNA methylation, right? So you're just going a little ahead of. So we have DNA methyl transferase inhibitors, right? Inhibitors lead to the demethylation of genome, thereby restoring the expression of methylated genes. And so these genes there are like 5 nucleotide DNA methyl transferase inhibitors. You know that because it's methylate on the cytosine, which is a nucleotide. So most of the and DNA methyl transferase are they going to nuclei and recognize the cytosine. So these are mostly the analog of cytosine, right? Like 5-azacystidine. Okay, these are the chemical name, and then this is the generic name azacystidine, or what does these are the drug names. 5-aza-2-deoxycysteine, dihydroxy 5-azacystidine, arabinosal 5-azacystidine, or commonly known as ARC, which is very much used in a lot of cancer patients. So these are just examples. So there's a lot of research has been done to block those transferases, because once you block them, then it means you can re-express the gene which are suppressed, and these are mostly tumor suppressor genes, right? And these are some of the structures. So you can see they are nucleotide. So this is a nitrogen base, and then for ribose, right? So these are like a nucleosides. Okay. So there's some example that 5-fera, which is just I don't remember what was it. 5-fera azacystidine. So these they are in number of clinical trials for treating solid tumors of hematological cancer cell. AzaCR was the first described DNA methyl transferase inhibitor almost like 40 years ago, and it still is used. So they are just to give you an example that these are the okay. In addition to nucleotides, analogs, they are non-nucleosides DNA methyl transferase inhibitor, and some of these are compounds but certain phytochemicals, things which are present in fruits and vegetables. And if I get time, I will go roughly touch on that. They have anti-carcinogenic activity, and because they have inhibitory activity for DNA methyl transferases, right? Compound created using modern techniques, we have a computer program where you can have an enzyme, you can see what binds and how it binds, and based on that compounds are discovered. And these are some of the compounds like hydrolazine, procan, procanamide, catechin 3 galate, which is a component ECGC, that's what we usually call, which is present in green tea, right? MG98, and these are again analogs, I think, based on ECGZ, if I'm not wrong. So hydrolazine is a drug to treat high blood pressure and arrhythmia, right? Its inhibitory activity towards DNA methyl transferase was first seen in patient who is systemic. It's seen as like systemic lupus erythematosus patient, right? In which is basically T cell leukemia. Same as procan is an anesthetic agent, it's often used, but now it's known that it also can be, you can do DNA demethylation. ECGC is present in green tea, right? It also decreases DNA methylation. Okay, and there's some other, more, sophisticated drug like MG98, rational design antisense deoxyoligonucleotide whose sequence is complementary to human DNT. So what happens that they bind to that gene which encoding the DNT and then destroy the gene. So these methyl transferases are not expressed. RG108 also this are designed on the basis of molecular modeling of the catalytic domain. MG98 in phase 2 clinical trial. So I'm not sure, it's a little, few years old slide, so maybe in clinical use by now, right? So this is all about the DNA methyl transferases. And then there are certain proteins. So I don't, want to go more in detail. These are just the protein which see the methyl cytosine and sit there, and there are a number of those. So these are they have all binding domains which bind to methyl transferases. There's nothing much really, and there are a number of binding protein which is bind to different kind of methyl transferases. And then again, I already showed you that the mechanism is that once it's methylated, the binding protein go there and recruit other protein, right? And then it recruit more methyl transferases, also recruit the deacetylases, and then it get more, methylation and deacetylation, and basically, suppression of the chromatin, right? Now, so this DNA methylation is also pretty much play a very important role in genomic imprinting, right? Anybody, any idea what is genomic imprinting? Okay, genomic imprinting is that, as we know that we have genes, and one gene comes from father, one gene comes from the mother, right? Okay, so we have two copies of the gene, right? Okay, only one copy is expressed, the other one is silenced, right? Okay, now the thing is that it actually, it like conserved that this gene is going to be expressed. So some genes which you inherit, which we inherit from our father, they express, right? And the mother side of gene is silenced, right? And some genes which are expressed in mother, they are expressed, but the father's genes are silenced, right? So it's not that, only one or the genes from mother side is silenced and the father side. So it's the imprinting. So we get an imprint from our parents. Which gene is going to be silenced and is going to be expressed. So genomic imprinting is parent of origin, specific allele. So the allele which is coming from which particular parent is like, have some kind of imprinting, right? So genomic imprinting is parent of origin specific allele silencing, right? Or relative silencing of one parental allele compared to the other parental allele. Okay, so this is again is occurred through epigenetics. Okay, so it is maintained in part by differentiated methylated regions within or near imprinted genes, and it is normally reprogrammed in the genome cell line. Okay, so genomic imprinting is an epigenetic mechanism of transcriptional regulation through which expression of a subset of mammalian gene is restricted to one parental allele. Okay. So, for example, in remember we have an X and Y chromosome, but in females we have X and X chromosome. So X chromosome is 2 times in females, right? So it means they should have 2 of everything, like whatever gene XX is chromosome is expressing, but that's not the case, right? So one X is expressed, but the other X is silent. Okay, so it is and it's silent because of the epigenetics, because methylated, highly methylated, or highly acetylated, so it's like become like a resonate, right? But what happens that you have 2 XX chromosomes. It's not that one is completely going to be silenced and one is completely going to be expressed. So it's happened like some gene from this X chromosomes are expressed, but the other one is silent, but some are silent on this X chromosome. So genes are expressed, but depends, it can be on both, right? But it can be a one functional X, but the genes are distributed on both, right? So you understand what I'm trying to say, right? Okay. Now, so just again, the same thing is explained here, that mammalian dose compensation by X inactivation, a process whereby one of the 2 X chromosome in female cells is somehow selected, usually more or less randomly, right? Although in special circumstances with the parent of origin basis, right? So again, it depends, which gene is coming, if the father gene is imprinted, like silenced, it means it's going to be silenced, there, right? The mother gene is not silenced, then that gene will be expressed. So if both chromosomes randomly selected, which gene is going to be, expressed and which is going to be silenced, but it's actually based on the genomic imprinting, based on their parental origin, right? So nearly all genes in mouse inactive X chromosome are fully silenced. Most genes in the human XY chromosome are also fully silenced, but there are some examples of X-linked human genes which escape inactivation and then deficient. And when deficient, these genes distribute to at least one clinical disorder, like Turner syndrome, right? So this is like some kind of disease with like abnormal growth of fatty linked to the breast development or something. So again, this is this just one example that this gene is expressed, and if somehow this is inactivated, then there's a consequences, right? Okay. So as I said, the express allele for imprinted gene is dependent upon parental inheritance expression of a gene. So it depends, whether the gene reside in a male or female in the past generation. So epigenetic means such as DNA methylation as CP sites control the imprinting of gene factor other than the sex of different parents could modify the imprint process, right? What it means that other things can change the methylation pattern, right? So although it's coming from father or mother, but then if there's a changes in the methylation pattern, it can change, right? Uh, and then these factors, right, can be like parental level of nutrition, stress, exposure to chemical and physical agent. So some of these changes can, because as we said that epigenetic changes are reversible, so they can remove the imprinting of the genes and then reprogram again. So it's really important. That's how there's a lot of emphasis on stress, that people should not eliminate this stress. So don't there's anything, try to minimize your stress, because stress causes all kind of chemical release and all that, and then you do not realize and you think it's okay, but it really hurts, and not to you but to next generation too, because it's passed on once the things get imprinted. Same as nutrition is important, and I kind of really scared the way our society is, especially in Pakistan. Everybody eating on road and restaurants and things become like a fashion. You have no idea what are you eating, what kind of oil they are using, what kind of meat they are using, what kind of spices they are using. Even the spices, I mean, like for example, if it's a cinnamon is 10 different varieties, right? And now the real cinnamon and then the other cinnamon, they have neurotoxin, they have hepatotoxin and all that, but because they're cheap, they look like cinnamon, and they have to make money, so they use the cheap product, right? I mean, they are. I mean, the thing is that there's another, this Ensil, there's star. What it called? I don't know. What you call in Urdu? This. What is it? Badiani, exactly. Yeah. So these are like also good for flavor and smell and all that, but they are like some are like a Chinese and other kind, which has like about like 40 times more neurotoxins in there, and they are very similar. So I mean, I don't want to extend my lecture, but the thing is that it's a habit, people doing it, but they don't know what they are changing. It's not just that you eat, taste, and everything is digested and get out. It actually affecting your gene too, because whatever they're doing, with stress or whatever they're causing, they're changing the imprinting, right? Okay, so here is an example. Okay, so here is, you see that there's are two genes. One is on and one is off, right? So that's in the paternal chromosome, and in this one, this is off and this is on, right? So we know that egg and sperm, they combine. So in this one, the gene one, which is on, will be expressed in this person, and that would be the maternal gene, right? Okay, and then in this, sorry, there's the paternal gene, and gene 2 will be from maternal. And it's the same example. It's just that we have this genes which actually, um, like in the sperm or egg, they get, we have only one copy, and they recombine, and they remember that which gene is silent and which gene is expressed, and then this, the whole thing is just um, kind of maintain in the adult form. Now, so why this is important? Okay, it's become very important because like to give you this example. So the first endogenous imprinted gene identified in 1991. So it's relatively new phenomenon. Well, for you guys maybe all, but I got my PhD in like 1988. So for me, it's a new thing. Okay, so what happens that homozygous IGF2 null mouse, right? Approximately 40% smaller than the wild type. Okay, IGF2 is an insulin-like growth factor 2. It's involved in the growth. Okay, null hypothesis. I mean, not hypothesis. Null means like they don't have gene. Okay, so this mouse is lacking the gene. Okay, so what would you expect? The mouse is going to be smaller. right? So that's why they were smaller when they born, right? Consistent with the known growth effect of IGF-1 2, right? But what happened then? Importantly, draw phenotype was also observed in heterozygous mice. Okay, so what are the heterozygous mean? We know that homozygous null mice means both genes are IGF genes are lacking. You probably understand why they are small, because none of these, we don't have IGF genes, so they're not going to be big animals, right? They're going to be small. Now in dwarf animals, they have they are heterozygous, right? So one gene is function, the other gene is silent, right? So it means if this gene should be working, so why they are dwarf? So then what happens? Then they discover that IGF gene is actually imprinted and expressed only from the parental, from the paternal allele. So it only comes from the father. Okay. So what happens that that so the thing is that if the mother gene is silent, right, and gene is and then the other gene is coming from the father side. Okay, is somehow the father side gene get altered, right? It's kind of blocked or suppressed. So what will happen? Even though you have a functional gene, right? But because from mother side is genetically imprinted, is not going to express, and other gene is somehow, it's supposed to be working, but somehow, if it is, if there's a mutation or something, then it's not going to work. So that in that case it's become very important that, it's not the only dominant and recessive that you have two genes, if not one expressed, the other should be expressed. But if other is genetically imprinted, so it's not going to be expressed, right? So that's what happens. So this is like a normal IGF-2, and this is the maternal, right? So maternal is not expressed. So these are normal. Okay, so even, you have a heterozygous expressing the normal, the mouse is of a normal size, right? Now the parental gene, okay, sorry, not parental, paternal gene, right, is a variant. Okay, so it means it's not going to code IGF-2, and this one is silent. So when a mouse carrying a defected IGF-2 is not going to express, is going to be still small, right? So this is just an example. So for example, if this is a cancer gene, right, it means if you inherit entire cancer gene from your father's side or mother's side, okay, and the other entire cancer gene like tumor suppressor gene, right, which is usually expressed, right, is silent from, depends on where you can inherit it. And if so you get imprinting, right, so one gene is not going to express, so you have only one. If anything happens to that, it means that that person has more chances to have that gene, tumor suppressor gene suppressed. Okay, second gene, actually the thing is that the interesting thing is that, the this mannose 6 phosphate receptor, or is also called IGF receptor 2. Okay, so it's encode the receptor, and IGF bind to the receptor. We talk about IGF. Remember, we talk about IGF-2 here, right? So this is the protein, and to bind to act, for this protein to function, it has to bind to the receptor, right? Induce the signaling by which the cell become bigger, or the animal becomes bigger, and all that, right? Now the receptor is actually it's let's skip the description, right? So we know that, IGF-2 is coming from the paternal side, right? Actually the receptor is comes from the maternal side, right? So it means the both mother and father, I mean, even the gene is active, right? So if there's a problem with the receptor, even though the paternal gene is working, that could still be a problem, right? Or even though, the receptor is working, but then there's no IGF-2, there's no point having the receptor, right? So there's what we call tug of war. So, and again, sometimes, the thing is that baby even is not born, and the mother and father is fighting, that whose gene is going to get expressed there, right? So this reciprocal imprinting suggests that the evolution of genomic imprinting may have resulted from a parent of offspring conflict to control fetal growth, right? So again, sometimes it happens that, you have parents which are very short, but their kids are very tall, right? Then again, depends, what happened to their genetic imprinting, what is expressed, what is suppressed, and sometimes then parents are really tall, and then they have a short baby. So and then so why how come our baby structure is only 5 feet and both parents are like 6 feet? So this is because again what is an IGF-1 and IGF-2, they're involved in the height. So it depends what is how the receptor and the protein is expressed from mother side or father side and all that. And um, all right, okay. So I want to skip some of these. So imprinted oncogenes and tumor suppressors, right? So imprinted genes are normally involved in embryonic growth and behavior development. Occasionally, inappropriate expression can affect expression of oncogenes and tumor suppressor gene. And what we get is like the loss of heterozygosity and uniparental disomy. So again, just to make this story short, it's the same thing. Like you get oncogenes and tumor suppressor gene. If they get imprinted from your parents, then they can play some role in oncogenesis, right? Depending what is active and what is not active, what is suppressed and what is not suppressed, right? And these are some of the examples like IGF-2, WT1, all these genes. They are known as imprinted genes. Okay. And the organisms known to imprint their genome is like seed plants, some of the insect species, and mammals, right? So remember, the seeds plants are very important because the quality, if you're making some kind of seed oil or something, so you need particular seeds to maintain the quality. That's where the plant maintain their quality because their genes are highly imprinted, and then they express only the one they're expressing, right? Okay, so these all about methylation. You want to take like 10 minutes break or maybe 5 minutes break? Okay, a break, and then we'll talk about histone modification. Any question you guys have? Sorry? Mhm. Is well, it's not the epigenetics is involved. It's not a main cause, because the like environmental effect, smoking 30%, obesity and 30%, but they use this mechanism. Yeah. So they're not because they are they are involved in the mechanism. So all these environmental factor and all that, smoking is modifying the epigenetics, okay, or maybe inducing the mutation, or maybe that kind of stuff. Yeah. You can, yeah, you can. I mean, you can be reversed. If you induce certain genes and there's been experiments done that you can transfer, differentiate. You take a different. There's a Japanese group who expressed, I think, three key genes in a differentiated cell that convert into a stem cell. Yeah. So because the genes which are silent, they just re-express. Yeah, something like that. Yeah. Mhm. It is focused. I'm looking at this. Usually I used to So what when in focus? So you guys are all doing some kind of cancer research or?
One of the side effects are that methylation the destruction of the gene and it can move in different positions. But how come like it is the hypomethylation? So how can it get
Well, no, for example, DNA where it's supposed to be methyl, right? So it's not to be considered. So when you open there and it's not one
by opening?
yeah, so there's a more susceptibility. So for example, there are regions which are so how do you find this cause? Learning something? There's no question. That's not the
Because we are following the cell biology that would be for gender document and it depends to get so it's somewhat the content is somewhat so we have an idea of the screen.
okay.
A bit easier to understand.
Okay. Okay, so people who are gone, if they are gone or they coming back? Okay, so we're going to start again. So now the second type of epigenetic mechanism is histone modification, right? Okay, so as I said earlier, that histone modification, it involves a lot of different covalent modifications, such as like acetylation, which involve acetic acid, methylation, addition of again methyl group, phosphorylation, which are the important transcriptional regulation. I mean, the histone modification is important in transcriptional regulation, and many are stably maintained during cell division, although the mechanism for this epigenetic inheritance is not yet well understood. Protein that mediate this modification, they are often associated with same complexes as those regulate DNA methylation. So because DNA methylation and acetylation go hand to hand, like you have DNA methylation and deacetylation in order to condense the chromatin, and then you have to go demethylation and acetylation, right? So they are most of it, as I gave the example that methyl binding protein recruit the deacetylase, right? So they are they're sometimes a part of the same complex. Now, Histone, they are like a basic, protein, contain a globular domain and a flexible charge amino terminal known as histone tail, right? You probably, is a basic histone structure, you probably learn in biochemistry class, right? Okay, so the histone tail is the one which go under colon modification, and it's like acetylation and methylation. So this histone acetyl transferase and methyl transferases, but it can also go into phosphorylation, ubiquitination, simulation, and ribosylation, right? So all these processes can modify the histone. So this is a typical structure of histone. So a histone is a complex. It's actually had eight subunits. So two of H4, two of H3, two of these are two H4, two H3, and then maybe two H2B and two H2A, and all these they have the tails. Now these tails, okay, as you can see, these are the sites for modification. Okay, so you can see these tails go under heavily modification. Okay, there are a lot of things like the green is like acetylation. So these are the sites for acetylation. Light green is deacetylation, uh, and then we have methylation. The blue ones are methylation, and red ones are phosphorylation. This purple is ubiquitination. I can see purple anywhere. Somewhere is purple, believe me or not. But oh, here. So these are four stones. So these are the other chains, right? So they get all these modifications. Okay. The enzymes which are involved, so lysine, these are the amino acids which goes into modification. So lysine often get acetylated, deacetylated by histone acetyltransferases and deacetyltransferases. Same as lysine also goes to methylation with respect to methyl transferases of methylases. Lysine also involved in ubiquitination, and the enzyme is ubiquitin ligase or protease, and serine threonine, they're involved in phosphorylation, arginine involved in methylation. So these are some of the amino acids which are involved, right? Basic structure of nucleosome, and I already explained, so I want to skip some of these things, and we already talked about how these are head, so these are the histone arranged, and these are details. So that's how a basic structure look like. So these are like 8 subunit, 4 top, 4 bottom, and then this is the DNA which is round, and actually it just so that it does not get unwind easily. So this is histone 1 is involved. So histone 1 is kind of put as kind of seal there so that it does not get rewind easily. So lysine, as I said, involved in histone deacetylation and acetylation. Histone methyl transferases involved in the histone methylation, which usually occur lysine and arginine. We have kinases, which perform phosphorylation, and we have histone dephosphatases, which perform dephosphorylation. So these are all basic. These are all basic that the acetylation is transfer of the acetyl group. So this is the lysine amino acid. So basically this nitrogen get acetylated, C double bond or CH with the acetyl transferase, and deacetyl is going to remove this acetyl group and do the deacetylation. So it's basic biochemistry, nothing much in there, and these are the tail of all these histones, which shows the site of acetylation, deacetylation. We know that if the hyle is acetylated, acetylation causes opening of the chromatin, deacetylation causes the condensation, right? And then can just skip this. Okay, so they're the histone deacetyl transferases. So these are important because what happens that we need deacetyl, deacetyl transferases, the one which suppress the gene, right? Because what happens when the acetyl group are removed, then gene gets silenced, right? So we have different kind of histone deacetylases, class 1, class 2, and class 3, and then within them there are certain numbers like 1, 2, 3, and 8 belongs to class 1, 4, 5, 6, 7, 9, and 10 class 2, and then class 3 is little different. Their numbers like this, instead of histone deacetylase, they are known as sertulin. Sertulin 1 to 7, they fall into class 3 because their mechanism of deacetylation is a little different. and they do not involve, their function is little different. So class 1, they are nuclear proteins, primarily regulate histone acetylation and chromatin structure, contain a deacetylase domain exhibiting like 40 to 90% identity in amino acid sequence to yeast. Yeast. Uh, okay, they are involved in cell proliferation in apoptosis. System 2, they are homologous to yeast, and they preferentially deacetylate non-histone proteins. Okay, class 2 are more specifically involved in regulating cell migration and angiogenesis, right? So class 1 involved in proliferation, class 2 involved in angiogenesis mechanism. They are localized both in nucleus and cytoplasm, whereas class 1 is mostly localized in in nucleus. They are 2-fold larger than class 1. The deacetylase domains are present within the terminal region, except the deacetylase of 6 contain 2 copies of the domain, one within the each N-terminal and C-terminal region. Deacetylase 11 has properties of both class 1 and class 2. So there are some exception. Now, homologs of yeast citrulline 2 they form a structurally distinct class of deacetylases because they use this enzyme, NAD, their own NAD dependent enzyme. They are found both in cytoplasm and nucleus. Okay. So deregulation of histone deacetylases activity by chromosomal translocation has been strongly implicated in aberrant gene silencing and promotion of tumorigenesis, especially in leukemias. So I'm just trying to skip so that I can cover some other topics. And these are basic, this basic concept. You understand acetylation, right? And deacetylation function, right? These are often present as a complex. The two major histone deacetylase complex, which called MSyn3A or new RD to RD. And these are really the acetylase is actually complex with a lot of proteins, and if anything goes wrong with any of these proteins, the enzyme get inactivated. And mostly what people are trying to do is trying to develop the inhibitor of deacetylase. So you inhibit the enzyme, you basically inhibit the whole enzyme activity. Okay, now, so we know that an inappropriate acetylation state of histone causes abnormal outgrowth and altered pattern of cell death, which leads to neoplastic transformation. So the histone deacetylases, they are overexpressed in several tumor cells. Okay, and the thing is that especially they are expressed like things are which are involved in tumor inhibition, like P53. Remember we talk about P53 is a tumor suppressor. So what happens that if you deacetylase that expression of P53 gene, then it means it's going to silence the gene, right? Same as P21 is also a very powerful regulator. It's involved in is a cyclin dependent kinase inhibitor, so it's involved in cell cycle, right? So these kind of some very key tumor suppressor proteins, they are basically a target of histone deacetylases. Okay, and then I just wanna, there's nothing. Okay, now, so there's a really growing interest to develop the inhibitors, and I'm sure some of you probably working. I know that sometime I talked to somebody who's working on DNA acetyl histone deacetyl transferases. So there are some like this is TSA, which is a well-known histone deacetyl transferase inhibitor. It was originally isolated from a fungal agent. This is a synthetic one, K22A. And they actually, all the inhibitors, they're classified into 4 different classes, right? So the first class is the short-chain fatty acid, okay, such as butyric acid and phenylbutyrate that are currently in clinical use. They're not identical because of high concentration. They require a millimolar amount to inhibit the enzyme. Now the thing is that you may have heard, but they are important. You may have heard that eating fiber is good for you, right? It says fiber eating is especially inhibit the colon carcinogenesis, and the reason it does is that when you eat fruits and vegetables, you get a lot of fiber in your gut, and your gut has bacteria which digest the fiber. Okay, and the end product is actually butyric acid. You make a lot of gases, right? So these gases are one of butyric acid is one of the gas, right? So this butyric acid is a very good inhibitor of histone deacetylase. So that's how eating fiber is recommended to decrease the incidences of colon cancer. Uh, so there are like some of these on that basis. Um, uh, I mentioned that you need a micromolar concentration in order to inhibit the enzyme. So on the structure of butyric acid, other structure are developed. Like this compound like the TSA, which is suberyl anil anilide hydroxymic acids, or it's called like a SASA, or oxampheline. These chemical names are sometimes tough to read. Okay, so these are actually basically based on the fatty acid. Okay, so there's the first class. Sorry, no, I was wrong. Sorry. So these are some drugs are designed the fatty acid. These are uh, on the second class is aliphatic compound that have hydroxymic acid. So they're based on this hydroxymic structure. So these are the derivatives of those. And then the third class, these are cyclic tetrapeptide. They have epoxy ketone or sulfohydroxyl group as a functional group, like trepoxin and FK228. So these are some of these HDAC inhibitors. And the fourth class is the benzamide bearing compounds such as MS27 or dash 275 or CL994. So these compounds, they are basically based on, see that these are like four different classes of histone deacetylases. It's just to give you an idea that there is like as we talk about the inhibitors for DNA methyl transferases, right? Are used same as they are inhibitors for inhibiting the histone acetylation. Okay, so they are not in, like promoting the acetylation, inhibiting the deacetylation. So again, these now class 3 is like a sertraline. So it uses like this NAD as a cofactor. So it does actually acetylate protein. So this is acetyl protein. So it's going to deacetylate into, remove the acetyl group, and the acetyl group is transferred to this ribose, and whereas nicotinamide is released. So this sertraline is actually they have a other role. So they involve in the in the regulation of various organs, and they are basically they are regarded as a factor for longevity. So their expression is actually linked to people who has those enzymes. Actually, it was done in Drosophila in a fly that when they express this enzyme, Drosophila live longer, and they found that uh, resveratrol, which is present in grapes, is kind of inhibitor for this. So that is linked that if people, well, this in your Western countries, they think if you drink a lot of red wine, which comes from the red grapes, it says high concentration of resveratrol, you might live longer. Okay, so I think I stop here, and then we just try to see if we can just let a little bit based on miRNA. Uh, Did you call him? I want to see the. I don't know how to. Need to stop share and. I know what to do, but I just don't want to do it. Just in case if I disconnect yeah, you stop share and then you start share the other one. Um, mm, क्या वो आ रहे हैं क्या? Hmm? You have to go the other number 4. Yeah, you already copied, right? Okay. So yeah, and then have to do. So what you do is stop sharing and then.
Yes, no, stop sharing, just skip it. So.
Okay. Okay. So maybe after half an hour, maybe.
Yes, I will be there.
Okay. Okay. So we're still there. It's not disconnected, right?
Yes.
Okay. Thank you. So I'm just skipping a lot of. You get the idea about histone modification, right? So you know what is epigenetics, is DNA methylation, and is histone modification, right? Now I'm going to spend some time, like not too much, but on mRNA, because it's also very comprehensive. I had a very comprehensive lecture on it, but then I condensed just to give some idea. You already probably know. So we know that don't know what is ribonucleic acid, right? So we have DNA base linked to the ribose, phosphate, so nitrogen base, phosphate ribose, right? So this is a simple structure of RNA. Now RNA, when I was doing, my master, we just know that there are three RNA, like mRNA, tRNA, and rRNA. Now we know that there are a number of different kind of RNAs too, right? So we have coding, messenger RNA, and then we have non-coding RNA, right? And which could be like ribosomal RNA or rRNA, tRNA, like tRNA, small nuclear RNA, or snRNA, small nuclear RNA, or SNO RNA. The interference RNA, RNAi, short interfering RNA, siRNA, or microRNA, miRNA. So we'll be talking about this miRNA, which plays some role in tumorigenesis, right? And we know how the RNAs are made. That we have a DNA. Part of the DNA start unwinding, and then it's going to replication. DNA undergoing to transcription, where it start making the RNA. It make a pre-RNA, and then part of the RNA are excised, is joined together and make a functional mRNA, which comes out in the cell, and then we have ribosomal RNA, which come present in nucleolus comes, and then we have tRNA. So ribosomal RNA with tRNA make this complete ribosome, which uses this mRNA to the protein synthesis. 50 years old biochemistry, right? Okay, so I'm sure everybody knows it. Okay, now the miRNA, so these are like short non-coding RNA, like they are about between like 19 to 24 nucleotide long, and then non-coding, so they don't code anything, and they are now regarded as a master regulator of gene expression. Okay, they are involved in post-transcriptional gene silencing of targeted messenger RNA. So what happens, because there are these, we know that in nucleotides we have complementary bases. So those nucleotides, this RNA go and bind to the RNA and actually silence the RNA, and that results in RNA degradation. Okay, so and they are involved in virtually all biological processes, such as like cell proliferation, apoptosis, development, differentiation, metabolism, immunity, neuronal patterning, stress response, aging, and cell cycle. So all the processes, they are regulated. They are strongly conserved on different in invertebrates, vertebrates, and plants, and they are probably not, about more than 1,000 miRNA which are identified in human, and more than 70% of miRNA are transcribed from the individual miRNA gene. So there is, before it was regarded as a junk gene. Now people don't know why they are making these RNA copies, but now we know that they have some function. Now, more than 50% of all protein coding genes are regulated by miRNA in mammalian genomes. Okay, so the way they are synthesized is, I'll just go briefly. So they are basically, we have like mRNA gene. It makes a pre-miRNA. So these are like mRNA and in a very long form, and these are then cut with this enzyme DROSHA and then DGCR8, which has sometimes their names are so big and long, depends like this is Drosha typically accentuated. Like DGCR8 is like George syndrome, critical region 8 protein. Usually people don't remember that, but just this short, right? And then so they are involved in silencing. So these are then cut off, and then pre-miRNA converted into pRNA. And then there's a transport mechanism. It's like exportin 5 and Ren GTP. This is loaded on these subunits and then transported out. So once it's out of the nucleus, then so then, and again, there's another example where the pRNA loaded, and then I mean it's kind of cleaved, loaded, and then transported out. When it's transported out, then it's further modification because still it's a double helix. So it's one of the strand is get lost. Okay. So again, it's the same thing that it comes out loaded, and then this loop is cleaved off. Now it's double extended, and then this duplex, the one of these strand is get cleaved, and you get only one strand, and this one strand is where is bind with certain proteins, which helps this miRNA to load to a target mRNA where it binds. So it binds, so it doesn't bind really very complementary, only few base bind, and then it's just enough to silence that miRNA there, right? So there's a nomenclature for mRNAs, which is and there's a database. Typically, what they have like a 3-letter prefix like MIR, right? For species of origin. So it's HSA prefix means human. MIR is the the miRNA. It's a Homo sapien MIR, right? And then after that they are assigned a number, sequential number, right? So like paralog sequence differ 1 or 2 positions. So like MIR 110A and MIR 10B, they are pretty much similar, right? They only few sequences are different. So mRNA and oncogenes. So cancer, we know that we have oncogenes and tumor suppressor genes. So again, what we have two types of miRNA. Okay, so two types of changes in mRNA are associated with cancer. And what it is a diminished augmentation expression of MIRs, right? Or increase. So some MIRs are, their expression is disappear, like they do not express during cancer, and some are they are expressed. Okay, so we have the similar term. We have onco-miRNA, okay, or onco-MIRs, right? So they are involved in tumor suppressor gene. Their levels are elevated, right? So again, because they are onco, I mean they are going to induce the oncogenesis, right? So their expression is increased. That's what they call onco-miRNA, right? But their function is, yes, they do, yeah. Okay. I'll explain you in a minute because in certain diseases there are specific mRNA which are expressed in elevated level, which can be used as a marker, serve as a marker, right? So MI10B is actually suppresses this HOXD10, which is a tumor suppressor gene, and this is mRNA. So this is an oncogenic mRNA, right? And this is just an example that where this gene is involved. You can skip here. So onco-mRNA, they're involved in silencing tumor suppressor genes. The expression is elevated. There's some example like MIR10B, MIR21, which is P10. We also talk about. There's also tumor suppressor gene, which is kind of inhibits dephosphorylate PIP3 kinase into PI. Is an antagonist of PI3 kinase. MIR71 to 92 involved in, and it's actually all differ like this, and it is a complex of all these miRNAs. They're all together. So here is P10, and we talk about that. Not P10 is block the activity of this, right? Conversion activation of AKT into phospho-AKT, which is involved in tumorigenesis. So P10 is a tumor suppressor gene. So if you inhibit, so if there's an onco-mRNA, it's going to inhibit the P10 or inhibit this expression of P10. So it will induce the oncogenesis, right? On the other hand, we have the same thing, right? So anti-onco-mRNA. Now their function is to inactivate the oncogenes, right? And some of the examples are MIR 143-7. So its target is RAS oncogene. MIR 26 target is PI3 oncogenes. So, and there's some examples of how they work. So these are certain miRNAs, and these are different sites. And so these all the pathways are involved in cell survival. So they are like inducing the oncogenesis. Cell is going to survive and multiply, but this oncoRNA, if they inhibit the activities of these uh, uh, enzymes or these protein which is coming from the genes, from oncogenes, so that's how they regulate it, right? And there are a lot of that. I apologize, you can't read it, but it just shows you the different tissues, right? And these are different kind of cancer of different tissues, and these are different miRNA which are upregulated, and some of the mRNA which are downregulated. So just by taking a blood sample, and you can and this has been happening to as a tool to identify cancer. So you can have a characteristic pattern which are expressed high, which are expressed low, these mRNA which are easy to detect, and on that basis, one can make some kind of get an idea that which kind of cancer a person is carrying or can be early detected, right? And again, the factor that affect mRNA expression is the same mechanism, because of it's work as a gene, like genetic abnormalities like mutation or frame shift or something. Anything happens to their gene can affect their expression. Epigenetic regulation, transcriptional regulation, post-transcriptional regulation, all these processes. So here is like chromosomal abnormality. So in this case, there's a point mutation, right? So it's going to affect the expression of this, right? So it means this is not going to cause the expansion of this mRNA, right? Whereas here is a chromosomal translocation, which is actually resulting in overexpression, can result of over. So this is like example of genomic abnormality, like a mutation or a chromosome translocation is exactly does the same thing what it happens to oncogenes, right? Okay, epigenetic regulation, the same thing. The regulation of mRNA expression remained largely unclear, because they are expressed, so they are promoter region. So these promoter regions can be suppressed through epigenetic mechanism, right? If they are promoter regions, has CPG islands, they can be methylated and they can be suppressed. Okay, they also have transcriptional regulation, right? That most important positive regulator of proto-oncogenic miRNAs expression are transcription factor encoded by proto-oncogenes. One of the best documented cases of this regulation involved MYC and MIR17 to 19 cluster, right? So these are involved in uh, so what happens? The MYC upregulate the expression of this cluster, right? So MYC, we know this is an oncogene, is regulate the expression of this cluster, right? So in this, the expression of this actually causes the robust angiogenesis and growth of tumor. That's how it induces the tumor, right? MR70 to 9 seems to exert this effect largely by targeting anti-angiogenic thrombopoietin 1 and related protein, right? So what happens, this RNA, because these are anti-angiogenic and the other protein which are anti-angiogenic, so they are destroy those proteins. So when these are destroyed, the process of angiogenesis starts, which is requiring tumorigenesis, right? So all these, there's has MYC, and it just shows that there's a regulation of P53 and MYC is a tumor suppressor gene, and MYC, so MYC expresses the various mRNAs which actually suppresses P53, whereas P53, results the suppression of some mRNAs which actually affect the MYC. So there's, and this is a tumor suppressor gene and this is an oncogene. So their expression is actually, regulated by these miRNAs. Okay, Post-transnational modification is when once it's synthesized, then there are processes which can also affect their activity, like phosphorylation, dephosphorylation. So once RNA transcribed into a protein, okay. So in this case, there are proteins which are modified, and this protein actually block the, for example, conversion of pre-mRNA, pri-mRNA into pre-mRNA, right? So these processes also involve their regulation, and then there are some of the protein we talk about which involve in carrying the protein from nucleus to cytoplasm, or involved in the recognizing the site of mRNA where it can cleave. So they get phosphorylated. When they are phosphorylated, their activities also change. Okay, so there's nothing much. It's just different examples of different things. So I want to stop here, and I want to actually go to some end the topic briefly. What is it? So you go and then minimize, right? Oops. And then I don't know if they are seeing this or not. We have to stop that one, right? Can you call him? Just I don't want to screw this up. New share. Okay. Now we need to we have to stop share.
So here,
New share, but we have to open the other one. Okay. Yeah. I just want to touch base on nutrition cancer. I think it's right or not?
it's all right.
Right. Okay. All right. All right. Actually, he helped me. So every computer has a little bit different mechanism. Okay. So I just want to touch this topic. I think which kind of help you understand some of the processes we learn about oncogenesis and all this nutrition cancer, right? So we know that you've probably heard this many times that let food be the medicine and medicine be the food, or you have heard that when diet is wrong, medicine is no use, right? When diet is correct, medicine is no need. So if you eat properly, you probably never need any medicine, right? But if you're not eating uh, right, then what happens? Then you have to eat medicine as your food, because I know that sometimes people use like 8-10 tablets in the morning, afternoon, and all there. So it's like there's their food, right? So nutrition cancer. So epidemiological studies like case control cohort or cross-sectional studies, they provide clues about between link between cancer and nutrition. As I earlier mentioned that 1/3 of cancer death in each year in US, and I'm sure probably more than that, like half of the death in Pakistan, can be attributed to nutrition and their other lifestyle factors like smoking, and process of carcinogenesis. Remember in very first lecture explained that it occurs over years. It's just not happening. So having a bad habit for a long time, actually that results in the carcinogenic process. So proportion of cancer estimated to be due to non-heritable factors is a big one. Like 90% almost like cancers, they're not inherited, right? They are from the environmental lifestyle, especially even just tobacco and diet can control about almost like 70% of cancer. And then again, still like certain infection, living hygienic, and some of the things like alcohol and or preventing yourself from occupational hazards like using masks and gloves when you're using some kind of chemicals and stuff like that can also prevent cancer. So a lot of cancers can be prevented if people take precautions. Okay, so when we talk about the epidemiological studies, there's some terms I want to explain. So usually people use like a case control study. So what is a case control study? Is the diet of individuals with cancer are compared with those of cancer-free control match for age, sex, and other key factors. So the two people, one is having a group having cancer, other group is not having cancer, but they're about the same sex, same age, living in the same condition, and then they will compare that what they're eating and what the other group is not eating. And then from there, one can find out that, okay, well, because this group is eating this compound all the time, maybe this compound is preventive, right? So that's how we will do case control study. Cohorts, so diet of different group of subjects are determined before cancer onset, and the incidence of developing cancer in each group are compared. Okay, so then like all the people are monitoring for long period of time, like select a group. Okay, we're monitoring your diet, right, and see what the incidence of cancer in different groups, and then on that basis one can hypothesize that which dietary factor may be more causing inducing cancer, or maybe preventing cancer. Okay, or the cross-sectional studies, right? The diet of different group of subjects are compared using the same measure at a single time point. Okay, so you're not doing a long-term monitor. This is like matching two different groups. It's just different groups are compared at a single point. Okay, just seeing that, okay, this person has cancer, this person not cancer, so just different group monitor and then compare, right? So this is a cross-sectional study, right? So there is a complex relationship between diet and cancer. Okay, dietary carcinogen naturally occurring, they are naturally occur and also added during food preparation and preservation. There are some of the dietary substances that can be enhancer of carcinogenesis, and some are provide like latency period between initiation and promotion, and then some are can be inhibitor of carcinogenesis such as antioxidant and phytochemicals. Okay, so diet contain both inhibitors and enhancer of carcinogens. So the food you eat can be either the safest and most powerful form of medicine or the slowest form of poison, right? For example, if you use excess oil all the time, over time what you gain obesity, and the obesity leads to a number of cancers. So it's like a kind of slowest poison, and eventually people die of cancer. Okay, so it's again, I don't know if you can see. So the thing is that you get nutrition mixture, nutraceutical, dietary fiber, phytochemicals, and all that. They undergo metabolism, okay, and then absorption, and they regulate a number of processes like inflammation, hormone receptor, biochemical inhibition, different pathway, hormone modification, DNA damage, and all that, and eventually they lead to changes in DNA, right? So nutrients ultimately have they have effect on genomes. Okay, so what we call nutritional genomics, right? Healthy aging. So if you eat like healthy food, you get healthy aging, and if you have a poor diet, then you have a poor health, right? And there's a big emphasis these days on there's a whole new class of research which called nutrition, precision nutrition. Not every food is good for everybody. There's something is because people metabolize food differently, and there's enzymes, and we have enzyme what we call polymorphism, right? In certain food, people metabolize efficiently. Some people don't metabolize efficiently. Some people they efficient in a way that become like toxic. So there are all kind of studies are done to recommend food for different people, right? So number one is energy intake. Like energy restriction inhibit cancer. You know that eating less is better. It extend lifespan in animals. So there are a lot of number of experiments are done that you give less food to animals, they live longer. So eating too much is does not really going to increase your life. Rather, it's going to decrease your life. Right? There's a positive association between overweight and cancer of breast, endometrium, kidney, colon, prostate, and others. Right? Overweight increases risk of cancer occurrence and decreases survival. Physical activity is inversely associated with cancer. We all know that, right? Okay. Nutrition, cancer, etiology. So some of the things which contribute in the cancer suppression or development are like fat, protein, soy, and phytoestrogens, carbohydrate like fiber and sugar, fruits and vegetables, and non-nutritive sweeteners. So other things like alcohol, coffee, and tea, and also the method of food preparation and preservation also play a role. The fruits and vegetables, and we know that they're different colors, and their colors are because these phytochemicals they have different phytochemicals which contribute to their colors. And for example, red color is mostly because of lycopene, and which is present like fruits and vegetables like tomatoes and tomato products. Pink grapefruit, watermelon, they have lycopene. Purple and red color, anthocyanin. That green color, they have sulforaphane, present cabbage, broccoli, and all that. So all these compounds, all the phytochemicals, they contribute to color, and then they have certain anti-cancer activity. Okay, so as I said, diet contain both inhibitors and enhancer of carcinogens. Animal studies also test that the effect of food and nutrition on cancer and provide guidance on epidemiology. So most of these studies are coming for animals. In fact, we know that high intake of fat or total saturated fat is associated with increases of breast, colon, lung, and prostate cancer. High-fat diet are also associated with obesity, which is linked with cancer of colon, rectum, esophagus, gallbladder, breast, endometrium, pancreas, and kidney. Animal fat from meat and dairy was associated with increases of breast cancer. It is found in one study. Whereas higher omega-3 versus omega-6 may reduce risk of breast cancer because omega-3 fatty acid, which is mostly derived from fish oil, they have anti-cancer property. Low-fat diet, like less than 20% fat, may reduce risk of reoccurrence of breast cancer. So, for people who already gone through this, got cured, they have to really decrease the intake of their fat. Total energy intake and physical activity. Total energy intake is strongly associated with breast cancer and post-menopausal women who already gone through menopause. If they have a lot of eating a lot, then they may at a higher risk, right? Physical activity has a protective effect. So women who spend about 4 hours per week in a physical activity had lower risk of colon reproductive cancer. So this is very essential for even for women, especially because women are very susceptible for breast cancer and cancer for other reproductive areas. And unfortunately, I don't know. Our women don't do much exercise. So it's important that you should really do some kind of exercise on a regular basis. And exercise doesn't mean there's a wrong concept that if you do walk, well, there is some benefit of walking, but when you do the exercise, you have to really sweat. That's where you're burning energy. If you don't sweat, you're not really doing much exercise, right? So obesity is a risk factor for all different kind of cancer. Among top is like a breast cancer, colon, endometrium, esophagus. We already talked about it. Now protein. The relationship of protein and cancer is not really clear. It's difficult to isolate effect of proteins. Increased protein diet are in fat and in fiber and decrease in fiber. Right, so it's not sure whether it's because of lack intake of fiber or it is lack intake of or increase intake of fat, because most of the meat comes with the fat too, right? Low protein diet seems to reduce the risk of cancer, while risk is increased by very high protein intake. Increased meat intake is associated with increased risk of colon cancer and enhanced prostate cancer. And again, you have to be careful when you eat meat. People use the barbecue and thing is getting in West, used to do barbecue and they eat kind of it, and we just really follow West blindly sometimes. So barbecue tonight, go and eat. They give money and eating this burnt food, right? So just to tell other people that we've been barbecue tonight, that's the only thing. Otherwise, there's nothing in there. Fiber, right? Two intervention trials evaluating the effect of fiber on polyps, which is kind of a colon cancer and adenoma, they actually failed to show the effect of high fiber diet, but generally it is recommended that people should use high fiber, even despite lack of conclusive evidence, because as I said, fiber helps in production of butyric acid and other small fatty acid, which are inhibitor of histone deacetylases. carbohydrate. Now, high glycemic index diet, they are associated with increased risk of cancer, particularly ovarian, endometrial, breast, colorectal, pancreas, and lungs. Uh, Limit processed food, okay, because processed food like jam, jellies, and things, they have high sugar, refined sugars, and emphasize whole grain and low glycemic index food. The sugar is really very bad. Sugar is what I call is really is a sweet poison, because the function of sweet is just taste. After you eat it, there is no other function, right? Okay, it's provide you energy, but the food you eat is provide you enough energy, right? The excess sugar you take, body has to get rid of it, right? It has to secrete insulin transport that sugar you eat comes into blood, it goes from blood into the there's only two places, either it goes into liver or it goes into muscles, right? It goes into muscle only when you're using muscle, right? With exercising, sugar will go into muscle because produces energy there, right? Our muscle has limited ability to store glycogen. It goes in liver, and liver has no option to convert the glucose into what? So you will convert limited glycogen, but liver can store only limited glycogen. It converts into fat, right? It makes triglycerides, and they which are transported out and stored where? In adipose tissue, right? So the adipose tissue grow bigger and bigger, right? So every time, just think about you taking a cup of tea, adding two, a spoon of sugar, which is just giving you taste, nothing else. But your body has to work to get rid of the sugar and make fat. So taking sugar really, not essential. If you start drinking tea or coffee without sugar, you're really not going to miss. I've been drinking tea or coffee more than 50 years without sugar, and I really enjoy it. Once you develop, then you really enjoy the tea and coffee, because you can really tell the taste of different coffees and teas. Otherwise, every tea is, doesn't matter what kind of tea leaves you use, they all same after adding milk and sugar, right? So here is the glycemic index food, right? So these are different food. So these are like anything which is like below 55 or below, they are low glycemic. High glycemic food about 70 or higher, they are high glycemic. So this is a table which shows that like things like ice cream, 60, or you use dates is about 1 or 6. And I mean, so there's things you need to avoid. So you can take low glycemic, because low glycemic, what happens is releases glucose slowly in blood. So your blood doesn't has to work that hard to get rid of glucose. And by the time it reaches the liver, you're probably working and doing some work, and you consume that glucose. So it does not really contribute much into increasing the blood glucose level. But the high glycemic food, when you eat, it suddenly increases glucose in blood. So you get a lot of sugar in blood, which insulin has to secrete and get rid of it, right? Fruits and vegetable found to be associated with lower risk in about 128 studies out of 156. So most of the studies, they have shown a positive association eating fruits and vegetables. Increased consumption of fruits and vegetables is associated with lower risk of cancer of the oral cavity, esophagus, stomach, and all these, right? Okay, evidence less strong with the hormone, related tumors like breast and prostate, because they are driven from a different mechanism. Okay, people who develop cancer tend to have low intake of raw and fresh vegetables, leaf and green vegetables, lettuce, carrot, raw and fresh fruits, cruciferous vegetables, flavonoids and lignins such as soy, grains, and vegetables. They are because they have phytochemicals which are their structure related to estrogens. So they are good. They play a part in lower. Eating them has a lower risk of hormone-related cancer, right? Fruit and vegetables, they also low in energy, good source of fiber and vitamins, good source of antioxidants like vitamin C, E, selenium, phytochemicals such as carotenoids, flavonoids. Okay, do not yet know what is a protective agent, so best to use food sources. Okay, what it means, you don't have to use the actual chemical, rather you just use fruit itself, right? So plant Based food may prevent cancer by inhibiting hormone-dependent steps in tumor formation and protecting genetic material from carcinogenic agents, suppressing free radical production, serving as a bulking agent to dilute carcinogens. Some of the fruits and vegetables, what happens? They bind some chemicals, and then some of the chemicals will be carcinogens, so they remove those from your system. The stimulator of physiologically active and anti-cancer enzyme. Beta-carotene supplementation in Finnish study was associated with the 16% higher risk of incidence of cancer. What happens that because beta-carotene, these are antioxidants, right? And the antioxidant and oxidant, they are actually reversible. Like some antioxidant can be oxidized, and then these smokers, because they smoking, so they generating a lot of oxidants, which kind of modifying. So they did not actually, they found actually higher risk. Heavy alcohol intake seems to enhance negative effect. However, diet suggests that high fruit and vegetable consumption, particularly carotenoids, tomatoes, tomato-based products, may reduce risk of lung cancer. Beta-carotene, vitamin E, and selenium at five times the recommended daily allowance was associated with significantly lower mortality from all cancers. So eating a lot of these will probably help you. Calcium, food high in calcium may reduce the risk of colorectal cancer, but calcium supplements may reduce the formation of colorectal polyps. However, there is evidence that calcium intake, especially supplements, is associated with risk of prostate cancer. So again, you have to balance out. So the good thing is that get calcium from food sources, not to take supplements. Okay, lycopene. Lycopene, as I said, is like a red color, pigment. Several studies have shown that tomato products reduces the risk of some cancers, but unclear whether lycopene is responsible. Absorption of lycopene is enhanced when lycopene vegetables are cooked or eaten with fat. So there's when lycopene is in the food, there's less evidence that supplements are safe and effective in cancer prevention. Lycopene supplement. Preserved meat and cancer risk. Some studies link eating large amount of preserved meat causes increased risk of colorectal and stomach cancer. We don't use that much preserved meat here, but in Western countries they use a lot more, which is high in actually salt, and they have it's filled with nitrates. Nitrate can be converted into in the stomach to carcinogenic nitrosamine, which may increase the risk of stomach cancer. So vegetables and fruit retard the conversion of nitrate to nitrosamine. So again, they provide some help. Cooked meat. Frying, broiling, grilling meat at high temperature create chemicals that might increase risk, cancer risk. Use techniques such as braising, steaming, all microwaving, all that, which does not burn the food. Okay. So naturally occurring carcinogen like aflatoxin in peanuts, saffron in plant oil, tannins in grains and grapes, benzopyrene formed by smoking meat. So these are some carcinogens which are naturally occurring. So you have to avoid eating a lot of peanuts. Usually they don't, but some peanuts contain aflatoxin. But naturally found in plants food like celery, parsley, figs, mustard, pepper, and citrus. All these things. Sometimes the farmers use pesticide, and some of the pesticides are carcinogenic. Okay, so these dietary agents like fruits and vegetables can suppress transformation and hyperproliferation, invasion, angiogenesis, and metastasis of many various tumors. So I will give you some examples. The exact mechanism is not found, but still, the recent studies actually has shown the molecular mechanism. And so again, so here, so common dietary compounds. You know that you heard about curcumin, which is present in turmeric, right? Resveratrol, red grass, peanuts, and berries. Genistein is present in soybean. Silymarin in milk thistle. Diallyl sulfide is present in garlic, I think, or elenium. I don't know what kind of fruit is. Allicin in garlic, lycopene, tomato, capsicum, chilies, diosgenin, fenugreek, 6-gingerol in ginger, ellagic acid, pomegranate, ursolic acid, apple, pears, and prunes, Enethol, anise, there's the starfish I was talking about, camphor and fennel, catechins, green tea, eugenol, cloves, indo-3, carbinol, and limonene, citrus fruit, like in limes, and dietary fiber. So these are very common and very cancer preventive. So if you start eating all these fruits on a regular basis, you will have a healthy diet. Okay, so we talk about some of these cellular pathways involved in carcinogenesis. Some of the genes are expressed like kappa B, AP1 activator protein 1. These are transcriptional factor. The serine protein kinases, STAT protein, which is second messenger, EGF receptor, cyclooxygenase 2, which involve in arachidonic acid synthesis, cyclin D1, histone deacetylases, chemokine, which is involved in cell migration, metastasis, proteasome, protein degradation, BCL2 involved in apoptosis, and caspase is also involved in apoptosis. So these are some of the targets where some of these fruits and vegetables work, right? For example, NF kappa B. So NF kappa B, what happens? You get the signal is present in cytoplasm. So NF kappa B is actually bound with I kappa B, get activated on phosphorylation. So the phosphorylated form, I kappa B phosphorylated, and the inhibition is gone, and then NF kappa B goes into nucleus and activate the gene. So it's a very powerful transcriptional factor. It's involved in inflammation. It's the key inflammation in almost like all cancers. There's the activation of NF kappa B. Okay. So again, what NF kappa B does is involved in like anti-apoptotic survival process, proliferation, tumor formation, metastasis, angiogenesis, inflammation, immortality. So all these processes are linked to, somehow linked to cancer, right? So the things which inhibit NF kappa B is like curcumin, genistein, capsaicin, resveratrol, green tea polyphenol. They all have direct target on NF kappa B. AP1 is another transcription factor, which is a combination of FOS and Jun is a dimer. So FOS and Jun, they make a dimer. They make AP1 is a transcription factor. So they get phosphorylated, translocated in the nucleus, and they control the gene of all these molecules. And so AP1 also control processes like differentiation, cell cycle progression, and apoptosis. So the things which directly affect AP1 activity is curcumin, caffeic acid, which caffeine, coffee, resveratrol. So directly interact with AP1, it suppresses AP1 activity. Resveratrol inhibit TPA, induce AP1 activation. Other oncogene we talk about AKT. Remember we talk about the receptor bind and then activate PI3 kinase. PI3 kinase causes formation of PIP3, which activate AKT, and activate AKT involved in inhibition of apoptosis and cell proliferation, whereas P10 reverses this process, right? So PI3 kinase and AKT is also an oncogene. So curcumin inhibit PI3 kinase, ECGC inactivates AKT, green tea polyphenol inhibits PI3 kinase and AKT. And the proliferation pathway we have is activation of MAP kinase, which also receptor bind to the signal growth factor bind to the receptor, activate, and all these processes they are linked to the activation of RAS, RAF, MAP kinase, MAP kinase, and then this involved in cell survival. So we have things like curcumin. You see, curcumin almost like very good. Haldi is very strong. In old days, people used to add haldi in milk and drink. And when we were growing up, and then people would say that then you think that there's something is not right. Now the science is proving the same thing, that people who was using things like about 100 years ago, they were so right, because they have seen the effect. And we believe on medicine, that you take the tablet, that's probably effective, but using these things on a regular basis could be pretty much effective too. Indole-3 carbinol, which is present in cabbage, inhibit different isoform of MAP kinase, resveratrol, reduces MAP kinase activity. ECGC, again the green tea, inhibits MAP kinase phosphorylation. And the transducer is SAT, which is they are also the present in cytoplasm. So again, they get activated, the kinases get activated, they phosphorylate is said, they make dimer, go into the nucleus where they regulate the gene. Most of the genes have the anti-apoptotic effects. So this is also involved in the oncogenesis. And curcumin inhibits Z3 phosphorylation, resveratrol inhibits the SRC3 pathway. Butene, which is present in cashew, suppresses Z3 phosphorylation. Capsaicin suppresses Z3 activity. So all these are very good, right? Now we have growth factors, right? We know that EGF, uh, um, which is inhibited by resveratrol. IGF-1 inhibited by the activity inhibited by green tea polyphenol. VGEF, right? Green tea polyphenol. EGF, curcumin, genistein, resveratrol, and catechins, they inhibit EGF activity. Chemokine, CXCR4, this is involved in metastasis, right? So what happens is that people, especially like when they develop the breast tumor, it can go to lungs, liver, brain, bones, different places. And this because these cells express these receptors, and then the inflammatory molecule which inhibit, stimulate the chemo, inducers, chemokines. So anyway, this we have things like curcumin inhibits CXCL2 mediated invasion and migration, reduce CXCR4 expression. Zerumbone inhibit against CXCR4 receptor, and CXCL2 there's a ligand which induces metastasis. Cyclooxygenase also involved in inflammation. Inflammation, actually, every cancer starts from inflammation. First is inflammation. If you do not control the inflammation, then it leads to the cancer formation. And what happens? We have arachidonic acid, which is present in the membrane. Arachidonic acid is converted into, actually released by phospholipase A2 activity, which is acted upon by COX-1 and COX-2, make all kind of sites, prostaglandin, cyclin, thromboxane, and all that. They have all like inflammatory activity. So there are things like resveratrol. They inhibit COX-2 expression in skin tumor, and nitrosomethylbenzylamine induces COX-2 esophagus, cancer, right? ECGC inhibit COX-2 expression. So some, resveratrol, ECGC can also reduce the inflammation. We have angiogenesis, where tumor actually sends signals and we draw blood supply from nearby, tissues. And there's all the processes. So we have like curcumin inhibit VEGF. VEGF is a strong stimulator of angiogenesis. ECGC catechin inhibit VEGF phosphorylation. Resveratrol inhibit angiogenesis. And we have cell cycles. So all these compounds like genistein, resveratrol, diosgenin, thymoquinone, which is present in black seed, right? Genistein, so they inhibit the cyclin D1 expression. ECGC upregulate these inhibitors, which kind of downregulate the cyclin-dependent kinases we talk about, right? And then SGE, this is grapeseed extract, reduces expression of cyclin D1 in lung cancer, right? And then so these are all different compounds. So that was, the diet is very good in controlling the different kind of oncogenes, right? Not only their diet is also important for epigenetic mechanism. So what we have, we talk about epigenetics, like we have histone modification, DNA methylation, and macro RNA. So these are the processes which involve in epigenesis. And we have compounds which can modify the histones and affect the DNA methylation. So we have, and some compounds they can do both. So like we have tomatoes, apples, citrus, and all these turmeric, cinnamon, soybean, all these green tea. They inhibit DNA methylation. Okay, and also it's compounds like garlic, grapes. They involve in histone modification. Right. So folic acid, you probably heard about is the folic acid, B vitamin. So it's actually involved in S-adenosine methionine is a methyl donor, and we know that methylation is an important process, right, for gene methylation. So folic acid so ECGC, which is present in green tea, right? It inhibits this DNA methyl transferases. And what happens that folic acid is act as a methyl donor, so it's inhibit that process. So I'm going too fast because I think it's getting late. Um, so SH is a potent inhibitor of DNA methyl transferase. ECGC directly inhibit methyl transfer, hypoxymethylation of tumor suppressor gene. They causes the hypermethylation. And again, you probably cannot read, but believe me, there's a big list of dietary agents which act as a suppressor of epigenetic processes. And I will give you a copy. And they also modify DNA acetylation. So these are the compounds which are involved in acetylation, diet and mRNA. Believe me, diet also regulates the expression of certain mRNA. We talk about onco-mRNA, which can act on tumor suppressor gene, and tumor suppressor anti-onco-RNA, which act on oncogenes. In curcumin, been shown to regulate like MR22, ECGC modulates, so then like MI16. Just to give you some examples, soybean, isoflavin, also involved in some mRNA, genistein, and so to conclude, choose a diet rich in variety of plant food, eat plenty of vegetables and fruits, maintain a healthy weight and be physically active, select food low in fat and salt, prepare and store food safely, do not use tobacco in any form. So, okay, so there was these are the recommendation for cancer prevention by American Institute of Cancer, and these are the recommendation by American Institute of Cancer Research, like almost like similar kind of recommendation. Choose a diet rich in variety of food, eat plenty of vegetables, maintain a healthy diet, drink alcohol only in moderation, which you're not supposed to drink. That's only for other people. Select food low in fat and salt, prepare and store safely, and then American Cancer Society, choose food that help maintain a healthy weight. When you eat away from home, choose food low in fat, calories, and sugar, and avoid large portion. Eat a smaller portion of high calorie food. Be aware that low fat and fat free does not mean low calories, and that low fat cake, cookies, and similar food are often high in sugar. Substitute vegetable food and other low caloric food for caloric dense food such as French fries. Okay, so don't use French fries, cheeseburger, ice cream, donuts, and all that. Rather, when you want to have some dessert, try to eat a fruit. Okay, so this is also just lifestyle, reduce alcohol consumption. Breastfeeding actually is a very good, in preventing breast cancer. Avoid obesity, which, being physically active is good. Eat more vegetable and fruit. Limit intake of lead. Avoid obesity. So here it is. Eat less crap, like carbohydrate, refined sugar, artificial sweetener, processed food. Eat more food, which is fruits and vegetable, organic lean protein, omega-3 fatty acid, and drink water. So I stop here. So sorry about this lengthy lecture, but I just wanted to give you a little bit of everything so that you understand that all the genes which are involved, tumor suppressor genes, and then we talk about all the epigenetic mechanisms, and then we talk about how diet is really important, regulating every process, right? Don't eat outside. All right, thank you very much. So if you have any question, I'm gonna give the PDFs handouts to you, so you have a copy. And if you have any question, feel free to email me. I'm probably leaving tomorrow, so nice, a pleasure knowing you guys, and hopefully we'll be in touch. Thank you.